Genetics is the story of what makes you, you. Who you are is written in the DNA inherited from your mom and from your dad. The combination of this inherited DNA makes up your genome, a blueprint for making you. Cloning means making identical copies, so a clone of an animal would share an identical DNA or blueprint with that animal. Although it raises many ethical issues and scientists have never cloned a human before, there was a case where scientists cloned a sheep named Dolly. Scientists took a donor cell from a sheep's udder, which is the mammary cell donor, and extracted the nucleus, which is the part of the cell that contains the DNA that could be used to make Dolly. They also took an egg cell from an adult female sheep and removed the genetic material. Then they inserted Dolly's DNA into the egg cell. This new fused cell started to divide, resulting in an embryo, which was placed in the uterus of a foster mother, where it developed into a lamb named Dolly. Since Dolly had the same genetic material as the original sheep whose DNA was used in the experiment, you would expect them to be exactly the same, right? Well, that is not exactly the case, and here is why. Scientists believed for many years that the DNA blueprint was the sole determinant of your characteristics. However, the relatively recent discovery of epigenetics shows that alterations can be made to genetic markers without changing the structure of the DNA itself. These genetic markers can be activated or inactivated in response to environmental factors. This is best seen when comparing individuals with identical DNA, especially clones. To make it easier to imagine, let us use the real-life example of monozygotic twins. Monozygotic twins are twins who are made from one fertilized egg cell which splits during embryonic development. As a result, they share 100% of DNA, making them nearly identical. In comparison, dizygotic twins, which come from two fertilized eggs developing during one pregnancy, share only 50% of the same DNA. Since the DNA of monozygotic twins is the same, you would think that they would be identical, but this is not the case. For the purposes of our example, let's meet twins Timmy H. and Jack Daniel. At birth, the two brothers seem nearly identical. However, as they go through life making different lifestyle choices, their environment alters their genetic markers, either activating or inactivating specific phenotypic expression. You can think of these markers as light dimmers controlling which genes are used and how they are used. As Timmy H goes through his life making good lifestyle choices, his epigenome turns on healthy genes and turns off unhealthy genes. Meanwhile, Jack Daniel, who throughout his life made poor lifestyle choices, turned on more unhealthy genes in his epigenome. This effect of environmental factors such as smoking, stress, unhealthy diet, etc. can have a drastic effect on the individual's epigenome and consequently their health. Meanwhile, making good lifestyle choices such as frequent physical activity, healthy diet, and avoidance of stressors can help you turn off the unhealthy genes and make the best possible you. As you can see, your genetics is not as simple as your exact DNA code. There are variables such as epigenetics that result in different expressions of the same genetic code. Unlike a computer where output equals input, we are influenced by our environment and our choices in how our genes are expressed.